you'll be a lot gladder if you use the right ladder and the right amounts. So today, some tips and tricks for using gel ladders, or more technically, molecular weight standards. So basically, if you walk into any sort of biochemistry or molecular biology lab, you're going to see things like this posted up, especially near like the gel boxes. These are basically mixes of proteins or DNA, depending on your application, or maybe even RNA, of known sizes that you can run on your gel electrophoresis next to your samples. And this is going to serve as a sort of ruler to tell you about the size of the molecules that you're trying to separate. So with gel electrophoresis, much more on this in other posts, but we are able to separate molecules like protein and DNA and RNA by size by sending them through this gel mesh. And so we use electricity to power them to swim through this mesh. The bigger guys, the longer guys, because they're typically unfolded or denatured, they're going to get tangled up in that mesh more, and so they're going to be traveling more slowly. And so when you stop the power box, well, now they're going to be higher up than the small things, which had an easier time slithering through. But how much farther? Where will they be in that gel? It depends on how long you keep that electricity on in the box. How it's working is that the molecules are negatively charged, and so you're then making it so there's a positive charge at the bottom, and they're going to be attracted to that charge. They'll go all the way through that gel if you let them. So when you stop it, they're going to be somewhere in there, and you need to, but you don't know like how big it is. If you just see a band, you need something to compare it to, and this is where this molecular weight standards come in. You want to make sure there are a few things when you're choosing one of these, because there's a lot of different options. One, the most important thing is that it needs to be in the range where your samples will be expected to be. So basically, if you think of a ruler, you would want to have it on some sort of scale where there's going to be bands that you can look in between. But unlike a ruler, it's not going to be evenly distributed. And so you can't just take the ruler and like move it up a little. You need to, when things run through a gel, things are going to run differently depending on the type of the gel and the buffer. And so if you're comparing to one of these marker charts, your thing might look totally different, but the samples that were in there were the same. They're just going to appear differently spaced. And so on some of the guides and stuff, you can even see how they'll look in different gels. You don't just want to make sure that your sample is in the range of those. You want to also make sure that you're choosing your gel percentage and things based so that you'll have a good coverage in the region where you're interested in. So if you do a PCR, you want to make sure that you're using a ladder in which your product sizes are going to be in the range of the ladder. When it comes to these ranges, there's going to be a lot more options for the nucleic acid, for like the DNA ladders, than for the protein ladders. Because typically for proteins, unless you're working on something really, really massive, or something really, really tiny, so they have like large molecular weight ladders and like peptide ladders if you're working with really big or really small things. But in general, the proteins that are found are going to be in the same sort of range. So when we talk about protein sizes, we're typically talking in terms of kilodaltons or KD. So you might see things written KD or KDA. Much more of this in another post, but this is a measure of the like protein's molecular weight. So how much a mole of it weighs in grams. So 1 gram per mole would be a dalton. A kilodalton is 1,000 grams per mole. So that a mole is 6 times 10 to the 23rd copies. So basically it's this way that we can convert between like the actual copy number and the amount that it actually weighs. We're not really talking about weight when we're talking about gels, other than that it's a kind of helpful way that we can talk about these sort of things. Other than talking, because we can't really talk about length, but that's really what happens is the protein unfolds and it's kind of slithering through. So its length is going to be determined by how many protein letters it is, the length in terms of amino acids, but our ladders are going to be measured in terms of like kilodaltons. So thankfully there's this helpful conversion where the average amino acid is about like 0.1 kilodalton or 100 daltons. So basically if you have a protein and you know how many amino acids it is, divide that by 10, you get a rough estimate of how big the protein is. So if you had like a 90 kilodalton protein or a 90 kilodalton protein, that would be about 900 amino acids. If you have a 900 amino acid protein, that's going to be about 90 kilodaltons. So you want And if you look at one of these typical ladder ranges, the one that I use a lot is this like Precision Plus. We'll get more into the various versions of it, but they have like pre-stained ones and then they have unstained ones. And you can see that it's going to cover this range from like 10 to 250 kilodaltons. And so most proteins are going to fall in between there, and so they're going to be covered by these gels. But then when you go and you look at DNA ladders, well, now you have a bunch more different options. Because if you think about it, DNA, like in our cells, is super duper 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 long. We're typically not like trying to run like bazillions of base pairs long DNA on a gel, but instead we're doing smaller pieces of it, such as pieces that we cut with restriction enzymes. Or pieces that we amplify with PCR. So we can have a bunch of different sizes though, from really, really tiny short things like oligos to really long products. And so we need to then have a ladder that's going to be in our range. It'd be impractical to have a ladder with bands of like all the way from like 10 to like 10 million, because think of all the little bands and how close together those would be. So that wouldn't be very helpful. So what's most helpful is if you have a ladder where the bands are kind of concentrated around the size of your expected products. So I mentioned this term KB. This is like kilobase pairs. When we're talking about DNA, we're talking typically in terms of base pairs because DNA likes to be double-stranded. So these two strands are going to be like hanging out together. When they're hanging out together, well, now we can talk in terms of base pairs instead of individual bases or individual nucleotides. There are, though, cases like when you're working with RNA or short single-stranded DNA, well, you might be talking in terms of bases. So for the DNA and the RNA ladders, they're often written in terms of like, so you'll see there's like 1 kb ladder. This is really helpful for just like general PCR. Then you have like 100 BP, 50 BP, 10 BP, like low weight, high weight, various things like that. When you see one of those like 1 kb, it's referring to the fact that these bands, most of them at least, are spaced out 1 kilodalton apart. So you have a band at 10, or again, you have 8, 6, 5, 4, 3, 2. So it's not like all of them are 1 kb apart. But then when you go to 100 BP, 100, 200, 300, 400, 500, that one makes more sense, et cetera, et cetera, et cetera. So you're going to have this, this is what the nomenclature refers to when it says like 100 BP. It's not saying that the top band is 100 BP. I was confused about that at first too, but instead the spacing is 100 BP apart. And as you can see, when you get to these higher weight bands especially, they're going to be closer together than when you have these lower weight bands. You might also, when you're looking at these, you might recognize, like some of these bands are going to be stronger, both for these DNA and RNA ladders and for the protein ladders. And this is going to help orient you to which bands are which on the gel. So because the bands can get close together, and also when they degrade, these have a tendency to degrade, especially the nucleic acid ones, then you can get like bands underneath it, like degradation bands, which you can confuse for the actual bands. Especially if you're using one of these like super duper high sensitivity gel imagers, they can pick up all those little degradation products and it can be really hard to tell what bands is what. So thankfully they have bands that are going to be stronger, and this is going to allow you to say, okay, I don't know what every single band is, like I can't tell what that band is, but I know what this band is. And if I know what this band is, well then the one above it must be 100. So for our protein ladders, with the Precision Plus ladders, the bands that are like these reference bands that have more are going to be the 25, the 50, and the 75. So you get really used to recognizing those on your gel. Speaking of these protein ladders, so these typically you come in like unstained or pre-stained. So unstained, like, well, your proteins are unstained, so you're going to be doing some sort of staining typically in order to see them, or some sort of Western blot in order to detect specific ones. If you have an unstained ladder, you can then detect it with just like when you're detecting all your other proteins with the Coomassie, but you're not going to see bands during the run. And you're also not going to be able to see them if you do a Western blot when you're transferring to a membrane. So one of the I typically use an all blue ladder when I'm doing a Western blot. so that because it's pre-stained, you'll actually see these bands when the gel is running. This is helpful because you can kind of visualize where things are in order to get the best separation. So we often say like run until the dye comes out, but depending on like where your proteins are, like the size of your proteins, you might want to run it a little more or a little less, and this is going to help you see that. It's also going to help you see during the transfer step that you got onto them, that the protein actually transferred from the gel onto the membrane. And so the pre-stained allows you to see that, and it's really helpful. Another helpful thing about these all blue is that the dye doesn't just let you visually see it, it also lets you see it like fluorescently. So if you're doing some sort of Western blot where your antibodies are fluorescent, you can actually scan it at like 635 is like the maximum, I think, and be able to see that. So I often use an antibody that's conjugated to a fluorophore that absorbs about the same wavelength, and therefore I can image them together and see the latter in addition to my samples. There's also like a kaleidoscope which has multiple colors and like a dual color. Those are going to make it easier for you to tell which band is which, even though like the bolder bands are easier to see. It's not like sometimes you can get them confused, especially if like your gel runs off a little, then it can be hard to tell which band was which, even if some of them are darker than others. And so the multicolor help with this. If you're trying to do fluorescence with those, then those different colors are going to have different wavelengths that they absorb, and so you need to look at the manual and see what that. For the unstained, if you're using one of the unstained in your Western blots, some of them, like the Precision Plus unstained, it's actually like SRP, or it's like streptavidin conjugated. So you can use like it has a strep tag conjugated, and then there's like you can use this streptavidin conjugated to a fluorophore or some like HRP or something like that. Basically, there's a way that you can detect it built in. And yeah, so for the nucleic acids, you just detect it just like you would any other. When it comes to colors and dyes and stuff, so when you're loading these onto your gel, they might come pre-mixed or in the loading buffer, or you might have to mix them yourself. It might seem like a pain, like you have to mix them. So with the protein, they're typically pre-mixed. With the DNA and RNA, they're often unmixed, especially when you're talking about the lower size ones. So this can seem like a pain, but it gives you a little more control over how much you want to load. Typically, you don't actually need how much they'd say that, like they recommend loading like 1 microgram per whatever, but maybe you only need 0.5, and then you can get away with, now you get twice as many samples. You want to also load a volume that is going to, like a concentration that's going to be in the range of how much you actually load of your sample. Otherwise, what's going to happen is you're just going to like way overload things with the ladder, especially if you're doing like a Western blot, or you're doing some sort of nucleic acid stain when you're using one of those like a gel dock machines that's going to try to autofocus. If your ladder is super duper strong, what's going to happen is it's going to like drown out the signal, and then you're not going to be able to see your sample, unless you like play around with the contrast a lot, or it might not. If you expose it long enough to see your stuff, then it's not, the ladder is going to be unreadable because it's just like a big blah. So yeah, so when it comes to the protein standard, it's like, so if I was just running like a gel, I would typically run like 10 microliters of a ladder. But if I'm doing a gel for a Western blot, then I'll do like 2 to 3 microliters, and that'll be plenty. When it comes to the nucleic acid, here it's going to depend on the concentration of the nucleic acid that you're loading. So if you're loading a really low concentration thing, you're going to want to do a lot less of the ladder. If you have a lot of concentration, you're going to want to do more of the ladder and things like that. When you're preparing these ladders, though, you might be having to pipette really tiny volumes if you're doing less, or even if you don't want to be freeze thawing these tubes a lot. And so what you can do is you can actually make a lot more than you need and then store aliquots of them, like single-use portions or a few use portions, but you don't want to be freeze thawing these a lot, especially with the small ladders. You can get bad degradation. Ladders can be really, really helpful, but they're not like, you can have something of the same like molecular weight or the same number of base pairs, but that can run differently. This can depend on, like, for the nucleic acids, it'll depend on like the base composition. For proteins, you can have the same length of a protein, or you can even have the same molecular weight of a protein, but they can have different, because they have different combinations of amino acids, and those amino acids have different shapes and charges and things like that. Even though you have SDS canceling out the charge, mostly you can still have some sort of weird effects. Bottom line, the composition of your sample is going to affect how it runs. And if there's modifications on the protein, that's going to affect things even more. So don't freak out if your product is not exactly like where you would expect it to be based on the size of the bands. Speaking of which, if you really need to know like exact molecular weights, you can do like a standard curve ladder type of thing with one of these, but you want to use the unstained, because the stained versions, then the dye can interfere with the running a little, so they recommend using the unstained if you're trying to do that sort of thing. But anyway, so your thing might not be exactly where you would expect it. And you can also have the problem where you can't tell the bands apart. And so if you want to know, is my product the right size, the best thing to do is to actually have a specific marker, like a positive control, that is the size that you're looking for. And so even if it's not the exact thing, if it's like nucleic acids, if it's something of the exact length that you want, because these are coming in like set lengths, so you have like you have 2 and 3, but what if your product was 2.5 and you wanted to make sure you had it at 2.5? You could have a 2.5 product. And so various controls are going to help you with that. This came into play a lot when I was in undergrad or in grad school, when I was studying microRNAs, which are around like 20 nucleotides long. And so I actually used standards, which was like another microRNA, to see if mine was about the right size. Also, because then I could like use a radio labeled one, because these aren't going to show up radio labeling. You can radio label these you can radio label ladders like T4 and polynucleotide kinase. Another option for getting a radio labeled ladder is to like transcribe some DNA or RNA, or no, transcribe some RNA or PCR, some DNA radioactively, and then like hydrolyze it so that you get different product lengths. So you can get like a 1 base pair step ladder like that. Yeah, so those were the basic things. Yeah, so expect that things are going to run a little differently on each gel, and they're also going to depend on the buffer. So your things won't look exactly like it looks in the picture, and that's totally fine. Your product might run a little higher or a little lower than you would expect. Don't freak out. Take advantage of these various things about the ladder, like the fact that you can use the fluorescence to see these bands on a Western blot, the fact that you can use the like HRP, streptavidin HRP, to see the unstained ones. So don't overload your samples. So keep it in the range of your samples, and also keep the like size scale in the range of your samples. Use specific designated markers, like if you're trying to distinguish between 2 different sizes, maybe you're doing a sequencing library and you want to try to cut out of a gel sizes of a specific pieces of a specific length, you can make markers and run those. Another thing is, like, it's helpful, I like to run ladders like at each end of a gel, and if possible like in the middle too sometimes, because what happens is that gels can like smile, and they can also just like run weird, and so you could have the different sides. And also when you're doing a Western blot, when you're blotting it, it kind of ends up tilted, and then if you were to try to cut it or something, you would cut bands off because it was tilted. And so what you can do is you can run multiple ladders, and that will allow you to then see the straight line and compare things better. But be wary if you're loading a ladder on each end. Make sure you can tell the ends apart. Like, so one of the things that's nice about having a ladder, like you always load the ladder in your first lane. Well, now you can tell what direction your gel is. But if you have it in the first lane and the second lane, and now you flip your gel over when you're putting it in the stain, and now you can't tell them apart, that could be an issue. So don't let that happen to you. And yeah, so that's the basics of using these ladders. There are also like specialized ladders for various things. But for the basics, these are like my go-tos are typically like the 1 kb ladder for like PCR reactions for like an agarose gel. I use like the 10 BP ladder for like small RNAs and things like that. Even though it's a DNA ladder, you can still use it for RNA. but I mean, like, the numbers will be slightly different, but that's why you have your like control markers. And then protein, like Precision Plus does the trick typically. There's like an XL version which has like a couple lower bands. Oh, speaking of the lower bands, not only do they have a tendency to like run off of the gel, but they also show up a lot fainter because the molecular weight is lower. There's going to be less of them and less stuff for the dye to bind to. And so they're going to show up fainter even if you have the same number of copies of them as you had of a bigger guy. Yeah, and I think that's the basics. Yeah, get used to using ladders. Love your ladders, but know that they're not like perfect. And be nice to them. Like, don't freeze thaw these a lot or they'll degrade. So pre-make some stock, freeze them down. Yeah. And for like the protein ones, they say to store them in room temp or in the fridge. We kept ours, at least the unstained ones, at room temp most of the time. In grad school, like it was okay, it survived. But yeah, better to keep them in the freezer. Especially with these DNA and RNA ones, definitely keep them in the freezer. Okay, good luck with your gels.